thank you also for inviting me to give a talk at the biology evenings. It's really nice to also go someplace abroad, even if I'm not abroad, but international talks are always good to have. And I hope I can give you some new information or some reminders of what you already know about stress and oxidants and antioxidants in plants and humans. And it's a time where a lot of us are stressed because it's close to Christmas, and but we are not talking about stress for shopping presents or something like that. We are talking about stress in our bodies and in the bodies of plants, and how plants and also our systems can manage these stresses. So let's see. Just have to check how I can turn the slide. Okay. So I prepared some graphics because it's easier than with a lot of text. So you probably know all of this, but it's nice to have a kind of easy introduction because it's already 7 in the evening. So plants provide us with a number of very good things like fibers, carbohydrates, water, different minerals, protein, fat, vitamins, and also antioxidants. And that's what we want to get into in more detail today. Before we do that, I wanted to show you a graph which connects plants and humans. And I think most of you know that plants use water, carbon dioxide, and sunlight to produce, on the one hand, glucose, so carbohydrates for their as their energy reserve, but also from carbohydrates then a number of other metabolites to grow and develop and flourish. And they also use photosynthesis, so the process of this, of what's shown here, to produce energy-rich ATP molecules for a number of different processes in their system. And the good thing for us is that they release oxygen because they don't need that much oxygen themselves. And they also release water during this process. And the water is good for us, especially nowadays when we talk about climate change and about cities getting hotter and hotter. So plants provide us with a kind of cooling effect by that. So that's a nice side effect for us. And the main effect for us is that we can get oxygen from the plants and chemical energy, as I've shown you on the last slide. And in our system, we use oxygen and chemical energy to also produce ATP, which we in our system use for a number of processes like digesting this chemical energy, or building different things in our body and metabolizing them. And we release energy as heat, of course, and we release carbon dioxide and water. So we can say that we are connected to plants in a kind of carbon circle where we release carbon dioxide, which plants can then take up again to perform photosynthesis and supplies again with oxygen. It's kind of a nice system which has developed over time. And during these processes like photosynthesis and respiration, which is very important for us because we need respiration to transfer electrons in the end to oxygen in our system. So we can get also these ATP molecules, which help us again to fuel all what we do. If we move or our nervous system, everything needs ATP molecules to function. But the problem is that in our metabolism and also in plant metabolism, there is kind of a side effect. There are oxidants which pile up, especially along these electron transfer chains, where the electrons are transferred from one molecule to another to gain these ATP molecules. And these oxidants are also often called reactive oxygen species, or ROS, or free radicals, or oxidants. So a number of names, but it's all the same. It's reactive or radical species derived from oxygen. There are also other molecules which can turn into free radicals, not only oxygen, but oxygen is the most famous one. And in our system, these ROS pile up, for example, in mitochondria due to respiration. They can also pile up in peroxisomes. These are cell organelles, both are cell organelles or cell compartments. Peroxisomes help with detoxifying these reactive oxygen species. And in our body, also inflammatory processes and immune responses can add to the reactive oxygen species pool. But on the other hand, you might have noticed that there is a small ROS sign down here, which is on purpose because small amounts of reactive oxygen species or other radical species are thought to be very important in cell signaling in our body and also in plants and other organisms. So it's not only that these free radicals or oxidants are the bad guys, but they also have important functions. And they tell our systems, for instance, to be aware, to produce antioxidants, to train the system to be more tolerant to stress or environmental changes. So they are not only the bad guys. And of course, we have a number of sources from the outside, so external sources for reactive oxygen or other radical species. Of course, UV light, I think you all know that. Sunburn, and what else can arise from that, like skin cancer and so on. Pollution, air pollution, water and soil pollution, industrial toxins, tobacco smoke, or ionizing radiation are the main sources of free radicals from the outside. But what's the problem with these free radicals or oxidants? A stable molecule is shown here. And if such a stable molecule loses an electron, it can turn into a free radical. A free radical is on the search for this lost electron. So it wants to gain back this electron, and it can take this electron that it's missing from another molecule, and this molecule might then be damaged, like a protein might be damaged, lipids might be damaged. And antioxidants, as shown here, are kind of the superheroes because they provide the missing electron to the free radical, so no other molecule is damaged. And this free radical can turn into a stable molecule again. This is why antioxidants are thought to be so important for our systems. And we are working with plants in Graz at our research group, and kind of the pet plant for all plant biologists. So most plant biologists is Arabidopsis thaliana, a small plant, which you can see here in the scheme. And we use it frequently because it's easy to grow and also easy to gain seeds from it, and it doesn't take too much space. So what we have done here, it's not my research, but it's research from my colleague Elmin Heinecke, Bernd Zechmann, and Maria Müller from some 10 years ago. They started experiments where they treated these Arabidopsis plants with different light intensities. So we might think, okay, plants, they love light because light provides them with sugars and they can thrive, but it can be too little light, like here it's too little, or it can be too much. And they were interested what happens inside the plant cell if there is too much light. So as you can see here, with this low light intensity, the leaves look green, they look healthy. We have one wild type and 2 mutants. And if we increase the light intensity to 300 micromole photon flux per square meter per second, you see that the plants start to react. We see it from the outside. Maybe, I hope you can see it, but there is a kind of purple streak here. and also some purple specks here. And this is what I call the plant puts on sunscreen. So it's not a severe stress for the plant, but it already recognizes something is not perfect. I better take precaution. And of course, it cannot put on sunscreen, but it will produce anthocyanins, for instance, which help to reduce the light stress on the leaf. And inside the plant cell, we have different compartments. So on the outermost edge, there is the cell wall. Then there is a very thin membrane. And inside this membrane, there is the so-called cytosol. And inside the cytosol, there are a number of cell compartments. And we have not depicted all the cell compartments here, but only the most important ones under stress conditions by reactive oxygen species and how they can be counteracted. So if there is high light reaching the so-called chloroplast, where photosynthesis takes place, there are stacks of membranes called thylakoids, and in these membranes, plants can generate these ATP molecules, and they can produce sugars inside the chloroplast. But if there is too much light, there are reactive oxygen species piling up, especially hydrogen peroxide. And what my colleagues found out during these experiments was that already after this slight kind of not stress, but a little bit of tickling the plants, they saw that hydrogen peroxide was accumulating in the chloroplast, but not in the other cell compartments. And at the same time, antioxidants, in this case, ascorbate or vitamin C and glutathione, also started to increase in this compartment. So the plant was able to counteract the amount of hydrogen peroxide under this light intensity. And when they increased the light intensity further, you see on the leaves that there is an increase in this purple stains on the leaf, and also some damage occurs. And if the plant is stressed too much, then the leaves start to deteriorate, so they get brown, they die, and they don't look happy at all. And inside the cell, you have more hydrogen peroxide inside the so-called peroxisome. Also here, there is a system to counteract this, but if there is too much stress and also photorespiration starts, then there is also an increase in hydrogen peroxide in the cytosol because it leaks from peroxisomes in the chloroplast into the cytosol. It might damage all kinds of proteins here. So also here, antioxidants started to increase, and also the enzyme catalase was more active in the cytosol and in the peroxisomes. And finally, when the stress prolonged even more, there was even hydrogen peroxide in the vacuole. And the vacuole is a compartment of the plant cell where the plant cell puts stuff like into a cupboard, things that are not so beneficial for the other compartments. And these compounds are stored there, or they are detoxified in the vacuole. So you see that plants can adapt to a certain type of or intensity or time of stress, but it's not indefinitely. So we can say, with a little help from free radicals, if there is moderate or short-term stress, plants and also our bodies can handle the stress. It's also beneficial because some antioxidants are produced and there is a shift. You see the scale here, more antioxidants than oxidants, and this is good for the system. But if the stress prolongs, you have high and or chronic stress. You see the balance is not points not in a beneficial direction, but it's more stress system, more stress, puts more stress to the system, and there might be damage, especially if this strong stress prolongs. So the goal is to be more on this side, and now let's see what we can do to get there. We have several antioxidants in our bodies that we can produce ourselves. Some of those are listed here. So all of these antioxidants we can produce ourselves. We can also produce glutathione ourselves. But what we cannot produce are these groups of antioxidants, which are here. And I've gathered some examples for you of sources of these antioxidants, like for example, vitamin C, of sources which you can easily get in our area. So in the area of Syria, of Austria, of Slovenia, and these are available most of the year, but especially in summer, it's not a problem to get fresh tomato, locally grown, or locally grown carrots. We can store carrots and we can store apples, so it's a kind of good source for us, and we don't have to ship it from another continent. So that was the idea. But of course, there are plants with much higher concentrations of vitamin C than the examples I've listed here. But maybe we can discuss about that later on. For flavonoids, I've already told you about anthocyanins. So anthocyanins are one type of compound which is part of the phenolic group of antioxidants, and there are more, and phenolic compounds are also found in apples, in grapes, and again in the three examples we already had here. And especially in, so for the anthocyanins, especially in the blue and red berries or other fruits, and phenolic acids and polyphenols more in grapes, especially in the blue ones. Then there is the large group of carotenoids. Some examples here. You might recognize pumpkin as a new example. And this is a winter salad. I've already also chosen because now it's winter, so it's in season. It's also easy to grow during winter time because it's not so cold in our area. And it's a good source of carotenoids. And there is even a grain which supplies some carotenoids. It's an ancestry form of wheat called einkorn. I couldn't find a decent English translation, so you have to live with einkorn wheat, and I'm sure there is a Slovenian translation for that. It's a very, very close relative to wheat, but it has smaller kernels, and the flour is yellowish, so you can see with your bare eye that there are some carotenoids inside. Then there are some sources of vitamin E. Vitamin E, or alpha tocopherol, is found mainly in seeds. That's why I chose pumpkin seeds and sunflower seeds, but it's also abundant in these other plants. And finally, I wanted to list some minerals. There are more minerals which can act as antioxidants, but we only have so much time. So I thought it's okay to stick to magnesium, selenium, and zinc as an example. And you see, if you look now at all the pictures with blueberries, for example, or with tomato, or also with carrot, you can cover a lot of different groups of antioxidants which can be found in plants. And for the blueberries, I also wanted to give you a hint. So what we buy in the supermarket or what we grow in our gardens, if we are so lucky as to have a garden, is an American type of blueberry. And it's not as rich in, for instance, vitamin C as the wild form. So the wild form, which you can find in European forests, is 10 times richer in vitamin C. So if you want to use blueberries, your homegrown blueberries or the supermarket blueberries, you just have to eat more. And that's not such a tricky task, I think. So after all these nice pictures, I also wanted to give you an overview in text form about antioxidant power of these different groups of antioxidants, just to see a difference or what they have in common in plants and in humans. For glutathione, it's one of the most important antioxidants to detoxify hydrogen peroxide, as I've shown before. It also protects against heavy metal stress. Plants cannot run away, as you know. They have to stay where they are. They only can try to send their progenies away, so their seeds or their fruits, but they have to stay where they grow. So they have to deal with a lot of environmental stress where we can run or hide from. And in humans, glutathione is important, for instance, for protecting the immune system and preventing inflammatory diseases. Vitamin C has similar positive effects on plants and humans. For us, it's very important in tissue repair. Most of you have heard of scurvy, I guess. So it's a very severe vitamin C disease or deficiency, so to say. And over thousands of years, sailors were even dying from vitamin C deficiency because nobody knew that they should eat fresh fruit and vegetables when they are on their journeys around the globe. And it's very interesting that already in the 14th century, some of the captains of some of the countries knew that they have to buy or get fresh fruit and vegetables for their people on board, but it never spread around the globe. So it took until the 19th century for systematic testing what causes scurvy and how it can be overcome. And it was not a nice experiment because the sailors were treated with, some were lucky, they were treated with lemon juice or sauerkraut. Both are rich in antioxidants, in vitamin C, but some had to drink seawater and some had to drink vinegar. So, but at least there was some information now on how to deal with that. Apart from tissue repair, which means for those who are not aware, so if you have a strong vitamin C deficiency, the gums start to bleed, so your mouth starts to bleed from the inside, you can lose your teeth, your hair, and in the end you can even die, so it's really severe. But luckily nowadays in our area, it's not a problem anymore. Then let's move on to phenolic compounds. For plants, they are also important for pathogen defense and herbivore defense because, as I've told you, plants cannot run away. So they tend to also produce compounds which may make their leaves taste not so good. So either other compounds are, for instance, soapy, but phenolic compounds are kind of stringent. So if you drink wine or if you've ever eaten a dark red or blue grape, which is not a table grape, but for winemaking, then you might remember that it feels like your mouth is getting dry and kind of velvety or something like that. And that's exactly the taste that some of the herbivores don't like. And it's also helpful against certain bacteria or fungi. And some phenolic compounds can also act as attractants to bees, for instance, like the anthocyanins. For our body, again, they are important for preventing inflammatory diseases, but also to regulate our blood pressure. If we move now to carotenoids, there are the most known, I think, are beta-carotene, lycopene, and lutein. Some of you might be aware about beta-carotene. It's the precursor of vitamin A, and vitamin A is very important for our eyes. So we should eat enough carrots or pumpkin or whatever contains carotenoids to protect our eyesight. And it's a UV light protection like in plants, but in plants is more like the carotenoids help to protect the plant main photosynthesis system, the chlorophyll system, from damaging by highlight, so by UV light stress. And carotenoids can also act as attractants, but I will show you that later. Also, some studies have found a correlation between carotenoid uptake in humans and cancer and cardiovascular disease prevention. For vitamin E, I've told you it's very abundant in seeds of plants. It's important for plants and humans for membrane protection. And I've listed some of the studies here. Most of, I think all or most of those are about human and antioxidants and how they work in our bodies. If you're interested to read more about that. And now I'd like to tell you a little bit about how we manage to visualize glutathione in plant cells. Or you can also visualize glutathione in human cells in a similar way. Because glutathione is a very small molecule. It's only 3 peptides, so it's 3 proteins linked together. And that's why it's not so easy to see with a microscope. So what we need is, we need to first prepare small pieces of a leaf or root, whatever we want to examine. Then we put it into fixative solution and prepare very, very thin sections. They are 18 nanometers thin, and it's very hard to imagine how small that is. That's why I try to explain it with a human eyelash, because we use a human eyelash to transfer one section from the diamond knife to the TEM grit or somewhere else where we want to do something. And the human eyelash is 0.15 millimeters, so 150 micrometers thin. And here we are a magnitude smaller, so 1,000 times smaller, and half of that. So it's extremely, extremely small. But the good thing is we have our antibodies. And you might recognize when I explain what we do, that it's similar to an antigen test for COVID, because our antigen is not COVID or a protein of COVID, but it's glutathione. Glutathione is in these small sections, and we can order an antibody which recognizes glutathione. It's specific for this protein. And if we put that primary antibody to the sample and wait for, say, 2 hours, then wash away the antibody that's not bound to the sample, we hopefully can see later on some labeled glutathione. So there is a link between the antigen, glutathione, and the primary antibody. Then we apply a secondary antibody, which recognizes not the antigen, but it recognizes the primary antibody. That's why they are linked here. And to see these very small structures in the electron microscope, a gold particle is conjugated to this secondary antibody. So it's very small. It's only 9 nanometers wide, roundabout, but it's visible in the electron microscope because the electron beam goes through the sample, and wherever it hits gold, the electrons cannot pass, but they have to go around. So there is a dark dot where the gold particle sits. You'll see it in a few seconds. Then the samples are contrasted with uranyl acetate and imaged with a sized library microscope. And we can even count the gold particles with imaging software. So we can say how many gold particles per square micrometer we have. And just to give you an overview of the plant cell, my colleague Barbara Kofler has prepared a very nice slide with false coloring of plant cell compartments. This is a compartment we also have in our cells. It's the nucleus where the DNA is located. Then we have peroxisomes, we have mitochondria, the chloroplasts in green, the plant cell wall, and just next to the plant cell wall, but not really visible, here is the cell membrane. And here we have the cytosol in pink. So the liquid where all the other cell compartments are located. Then I've shown you already a vacuole in a scheme, so the kind of cupboard of the cell where things are stored which are not needed at the moment or are not so beneficial for the plant cell. And sometimes there is extracellular space between the cells. In this way, it's hopefully easier for you to recognize also the cell compartments in this image. This is an immunolabeling image where you can see chloroplast, mitochondrium, another chloroplast, and the cell wall here, starch grains, and another chloroplast here, a part of a vacuole here, and cytosol all around here. So cytosol is not empty liquid, but there are all kinds of substances inside and also some smaller cell compartments. And if we enlarge this part, you can see the gold particles. All these small black dots are gold particles bound to glutathione. And the handy thing is that there are also antibodies specific to other antioxidants. So you can also visualize others. In this case, just for those who want to know, it's samples from maize. And the pictures were taken by our current PhD student, Leah Fladenhofer. In another project, we looked at a highlight again, but not as high as in the first example I've shown you. We chose an increase in light, which is not damaging to the cell. So from the first experiments with Elmin and Bernd and Maria, we knew that 1,500 and 700, that's too much for the plant cell, but we thought, okay, 500 micromole photon flux density, so irradiation might be not too much. And indeed, you can see there is no damage to the cells. They are intact. This is the lower light intensity, the control light intensity. There is no real difference in the cell compartments. They all look intact. But we could see when we were counting the gold particles that there is an increase in glutathione when we apply a higher light intensity. So we think one can train plants to withstand a little more stress. And interestingly, the same effect was found when we applied shade. It was not shade by putting an umbrella or something, but shade in the sense of providing the plants with a low rate of red to far red light. So more far red light than red light. And this should resemble kind of a leaf shaded by some other leaves, for instance. And you can see that the effect is quite similar. If you put a leaf into the shade or into a slight increase of light, it increases most likely hydrogen peroxide. We did not measure that in these experiments, but we found an increase in glutathione in both treatments, and that was very interesting to us. And I've promised you that I also show you an example for antioxidants and especially carotenoids as attractants. And this was a project together with our collaboration partners in the Netherlands with the University of Groningen. And they had very nice experiments where they looked at differently colored flowers, as you can see here. This is physalis and different types of petunia. And the plants were kept under control conditions on the left side and under sulfur deficiency. So plants were not given enough sulfur. And it's interesting to see that only the carotenoids, so the yellow flowers, and also a little bit this reddish petunia show kind of a pale color and also disturbance in their reflectance pattern. So this reflectance pattern was measured by the zoology colleagues because they can then correlate these reflectant patterns to the attractiveness of this flower for a bee. And if it looks like this, it's not very attractive for a bee. Even worse was the case for rapeseed. You see a normal rapeseed flower here and a sulfur deficient rapeseed flower here. And this is the control reflectance pattern, and this is the sulfur deficient reflectance pattern. So the flower is pale, but it's also much smaller. And it's a real problem for rapeseed because bees recognize smaller flowers as pollinated. So they will not visit the flower. And it might be a real problem for agriculture to actually have sulfur deficiencies and small pale flowers because bees are not interested then in visiting the flower. They will just pass on to some other nice flower with normal size and the normal reflectance pattern. Of course, we also wanted to know if it's really the carotenoids or if it's some other compound which is decreased, but we were pretty sure it's carotenoids. And you can see here from HPLC measurements that there is indeed a decrease between the normal grown plants and the sulfur deficient plants. And as a last project or cooperation or example from our group, I wanted to show you something more applied. So there is a research station quite close to our university in the south of Styria, in Wies, and they grow different types of vegetables and fruit for the local farmers to test which varieties of, say, tomato, potato, sweet potato, whatever is best suited for the local soil and for the microclimate in our region. And in this cooperation, they wanted to try different kind of sauce tomatoes. We might have recognized it from the shape of the tomatoes. They are all thought to be well suited for preparing sauce or preparing ketchup. And there was a blind tasting in Wies for taste. And all of these varieties ranked quite well or were exceptional in taste. But this variety was ranked comparatively low by the testing team. So these are the names of the different varieties, and one might stand out to you because most of you may know San Marzano. It's an Italian tomato, and it's very famous for being a good sauce tomato. And if we look how much vitamin C in these varieties we can find when we harvest them in a ripe stage, you see that there is not such a big difference, but this one is outstandingly lower compared to the others. It's not extremely low, but it's significant. And it's not always the same if you look at another group of compounds. So if we switch from vitamin C to the carotenoids, you see that San Marzano performs almost as well as the others. And there are 2 other varieties which perform not so good. And for beta-carotene, there is again another pattern. There is red cavern standing out. It was bred exceptionally for having a high beta-carotene and lycopene content. So it performed, of course, really well compared to the others, but the taste is not so good. And our idea is to find Styria's next top tomato among these varieties, one with exceptionally good taste and at the same time with a good combination of nutrients. That's why we also checked vitamin E or alpha-tocopherol, and you see it's again another pattern. So we will go on in analyzing different compounds, also compounds which are important for the taste, because vitamin E, carotenoids, and so on, they are not so, vitamin C is not really the compound which gives the taste we like to tomato. And that's why it's important to also test these other compounds. So I hope it was not too fast for you. I tried to be slow, but I tend to run in the end. Some conclusions. So we have seen that there are internal and external sources of oxidants. I've shown you that it's important to have a balance between oxidants and antioxidants, and it's beneficial that the balance is more in favor of the antioxidants, but it's not good to not have any oxidants at all. I've shown you that antioxidants are multifunctional, and I've introduced you to a method called immunogold labeling, which detects antioxidants in cells. And last, I've also shown you some examples, among them the search for the best tomato variety, and we go for taste, yield, and antioxidants in that case. So, take-home message for us humans is eat it all. I say that because it's really important to not peel an apple or a carrot, and eat raw and cooked food, because some of the ingredients or some of the compounds are easier to take up by our bodies if they are raw, and some are more easier taken up if they are cooked. And with that, I'd like to acknowledge a lot of people. I will not show you everyone separately because that takes too long. This is our team in Graz. It's not the entire team, it's the team involved in the experiments I've shown you. We have even more people involved in other projects. These are our cooperation partners from the University of Groningen. This is our cooperation partner from the Hungarian Academy of Sciences. Claudia is our partner in Wies with the tomatoes, and Christian is our new cooperation partner in Klosterneuburg also for tomatoes. I'd also like to acknowledge the funding agencies and especially our students, because they perform most of the work. So I think it's good to thank them for all their enthusiasm when they do their thesis. And with that, I'd like to wish you happy holidays. If you are interested in more of our research, please check out my ResearchGate profile or our stress and cell biology webpage or Forschungsportal of the University of Graz. There you can find all what we are up to. And I thank you for taking the time.